Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in creating a better tomorrow for all of us and our pets. Today, we have the honor of being joined by Ashley Kalinauskas, who is the founder and chief executive officer of Torigen Pharmaceuticals, which is a really interesting company. They're dedicated to researching and developing these novel immuno-oncology products and services specifically for the veterinary market, with a focus on really unique, innovative autologous cancer vaccines for pets. Torigen is a startup. It ultimately resulted from Ashley's graduate thesis project at the time at University of Notre Dame. There she was working on her master's in engineering, science, and technology entrepreneurship in collaboration with Dr. Mark Sukow at the time at Notre Dame, who was working in this really fascinating area of autologous cancer vaccines. Ashley started off at University of Connecticut. There she got her undergraduate degree in veterinary pathology and pathobiology, and then hasn't looked back and is on a really interesting entrepreneurial journey that potentially can affect millions of our dogs and cats and other pets. Honored to have her. A lot to talk about. Ashley Kalinauskas, thank you so much for taking time to come on the show.
Yeah, thank you. I appreciate it. Thanks for having me today.
Yeah, no, it's an awesome topic that we don't normally get a chance to go into, so I'm really excited about it. Would love to start off, sort of we typically do by giving you the floor for a few minutes just to further introduce yourself and talk about you. I'd love to hear everything, sort of the background story, where you grew up, how you got interested initially in obviously veterinary pathology and pathobiology, and if you could take us up sort of to sort of 2013-ish time frame where you first connected when you're at Notre Dame with Dr. Sukow. I think that would be a great way to set the stage for everything we're going to be getting into with the company.
Yeah, absolutely. Thank you. And a little bit about me. It's always hard to talk about yourself, especially being able to see the journey from where I've come from as well as where this business has come from. But I grew up in Connecticut, and for me, science has always been a love and a passion. And, I really think back to kind of like what first got me interested in science. And I think about, high school biology and just, a book of viruses and vaccines that was on a shelf that I took home one weekend. And I went to a cheerleading competition, being a really competitive cheerleader. And then I fell in love with being able to think about, the creation of different vaccines for these, viruses. And that led me to major in pathobiology at the University of Connecticut, where I was working in a novel vaccine lab specifically for, it's called PRRS, porcine reproductive and respiratory syncytial virus.
Yep.
And, was an athlete at the University of Connecticut too. So kind of had a lot of things on my plate in order to juggle. But at the end of the day, I was always focused on how can I take what I'm working on in the lab and actually make it see the light of day? What would it take in order to commercialize something that's really high science and be able to actually help, in the case of the PRRS vaccine I was working on, help millions of pigs that, could potentially get this devastating virus that leads them to die. So it's being able to have not only that animal welfare piece, but also that business acumen in order to combine the two. So we started taking business classes as well. So ended with a business minor. And I really found my love for that connection and intersection between science and business, because I think that there's sometimes not enough people who can really talk about the next steps in order to commercialize something that they're working on. And, that doesn't mean that everything that's worked on in the lab needs to have a commercialization route, but if it can, someone needs to ask why not, and what would it take in order for us to get there. So that led me to Notre Dame, where they had a really innovative master's degree program where you were working in the lab of your discipline, but then you're also taking all MBA classes at the same time. And instead of a traditional scientific thesis, you're saying, what am I doing in order to commercialize the technology that I'm working on? So it was really kind of checked all of my boxes, because I was like, oh, I was trying to do this in undergrad, but I didn't know where to start. So now being able to continue that really led me into a vaccine laboratory, again,
Mm-hmm.
my specialty. And then instead of your traditional virus vaccines, it was how can you make cancer vaccines? So the technology that I worked on under my PI, Dr. Mark Suko, was the commercialization of an autologous or personalized cancer vaccine that utilizes the patient's own tumor cell as the antigenic backbone for the creation of these vaccines. So and the lab had worked on proving this out for prostate cancer, ovarian cancer, melanoma, mammary carcinoma, squamous cell carcinoma. We were able to have a lot of different information, publications, and patents, but it then became that gap between, well, what's next? How do we take this and actually be able to make a difference? And when you think about, what it would take to bring this into humans, it was, 10 plus years and $100 million. And there were some previous high profile failures of personalized cancer vaccines in the market. So, a lot of that kind of started with the reflection of, okay, well, why did those cancer vaccines fail? And, unfortunately, how we, treat humans with cancer is we go back to what standard of care is. And standard of care has been established, for decades of chemotherapy and radiation therapy and different protocols in order to help advance it. And while immunotherapy is amazing, it still is pretty new in regards to the overall ability to get these commercialized products out there. So the first autologous cancer vaccine that was commercialized was a dendritic cell vaccine for men with prostate cancer from Dendrion, a company or the product Provenge.
Yep.
But there was another kind of really similar to our tech failure that failed phase 3 randomized double-blinded controlled clinical studies, because they had to treat patients that on average failed 20 rounds of chemotherapy and radiation therapy. So what functional immune system did those patients have?
Right.
And then, even when you look at the Provenge clinical data, they were measuring overall efficacy for about 60 days. And, you're dealing with very, very end stage humans with cancer by the time that, immunotherapies can be considered. Now, with the creation of checkpoint inhibitors, which are monoclonal antibodies that really help drive that immune response earlier, there's been a lot more positioning earlier and earlier after the cancer has been diagnosed. But there's still this gap between how do we get to the patient that has a functional immune system and actually being able to treat them. And that's really where the veterinary market kind of, again, checked our boxes in terms of the ability for us to advance this technology forward. One, you have a chemo-naive patient. You, two, have typically very early stage disease by the time that patient is coming in with a lump or a bump or not feeling good. And, very rarely do those patients continue onward for chemotherapy and radiation therapy because of cost. So for us, with 4 million dogs and 4 million cats being diagnosed with cancer each year, with 50% of all dogs over the age of 10 dying from cancer, it became, why not us to be able to help these veterinary patients?
Mm.
So together, Mark and I launched a startup company, Torigen. We worked on it kind of over the next couple of years, more so like kind of in the background as we were collecting additional safety data. We were collecting some initial efficacy information and data. And then in 2017, started a laboratory at the University of Connecticut in their technology commercialization program, where we set up Torigen as a little tiny company, as me as employee number one. And we started to grow from there and haven't looked back. So from employee number one to now I have over 30 employees, over 16 million in capital raised. We've really been able to make a difference in the lives of these pets and their owners in order to provide this personalized cancer vaccine. And to date have treated just over about 4,000, if not 5,000 pets.
That's awesome. No, that's really awesome. And I don't, we're neither a dog or a cat family. We're a bird family, but we can get into that topic a little later. But yeah, looking at these numbers, I mean, you mentioned the 4 million cats and dogs diagnosed, and I think we're talking about 65 million U.S. households that own a dog, about 47 million that own cats. The cancers that they get are slightly different. And I know that, you've done work and I had to look up sort of what hemangiosarcoma or hemangiosarcoma was all
Sarcoma.
about. Yeah, that's sort of the dogs get more sarcomas and carcinomas. It seems like cats get more lymphoma. But can you say a little bit more about, because the market is obviously huge, and at the same time, as you were mentioning, per the sort of the chemo naive stuff. We don't always think about it. I have friends that sort of work in the sort of the old school vet pharmaceutical industry, but families think about these pets, the dogs and cats, like children. I mean, they usually think of, when we're talking about what we're picking a therapy. No, it's not. It's not for me. It's for something a lot more delicate. Talk a little bit about this. I think this is an extremely important point, so people understand sort of the scope here of what we're dealing with.
Oh, no, totally. I mean, we don't even refer to pet owners as much as we refer to them as pet parents. I
Yeah.
mean, right behind me is my dog, a
Yeah.
painting of my dog. I mean, he is my child. He's my baby. And I would, walk across the earth in order to do anything in order to help him. And with such a huge proportion of pets being diagnosed with cancer, it then becomes, well, what would I want to put my own dog through? Would I put my own dog through chemotherapy and radiation therapy? All right, well, here's a few things that we know. We typically know that chemotherapy is not as bad in dogs as it is in, what we typically think of in humans. Very, very, very rarely, if not, ever do dogs lose all of their hair being on chemotherapy. Yes, they'll be sick. Chemotherapy is meant to stop cellular replication, which is what our body needs in order to divide and grow. But the side effects are a little bit less pronounced in dogs, and it's very, well tolerated. But the cost of being able to treat your pet with chemotherapy starts at about $5,000 and can quickly work its way up to $20,000 or more. Not only that, the time commitment needed in order to treat your pet. Like, we have to be cognizant of these owners that want to do something, but maybe don't have the time to bring their pet an hour away 3 times a week. So it becomes well, how can we really bring immunotherapy up to the forefront? So when it comes to understanding, like, chemotherapy, radiation therapy, immunotherapy, we also have to understand kind of like the responses that we're aiming and trying to build. So with immunotherapy, the goal is to have the body finally recognize what went wrong in order
Mhm.
to be able to stimulate a response against it. So when I think of cancer, I think of, there's kind of hot and cold tumors. A hot tumor means that you have a lot of of white blood cells or lymphocytes inside of that tumor that are no longer doing their job. The tumor has found a way to turn them off and they are no longer active. Or a cold tumor means that white blood cells or lymphocytes have actually been blocked out of that microenvironment. And in that case, we need to start flooding into there and we need to be able to create a response. Well, with a cancer vaccine, the goal is, can you prevent that localized recurrence of that tumor or metastasis of that cancer after a surgical resection? So can you be able to disrupt that microenvironment enough, capture those mutations, and be able to present them back? And, I think that with a therapeutic approach like ours, it kind of, again, it's a short treatment protocol, once per week for 3 weeks, of a series of subcutaneous injections to help stimulate that immune response. And the price is typically about half the cost of what an initial chemotherapy protocol can be. So it's affordable, it's accessible, it can be done at any general practitioner's office, and it can really, help induce that immune response to help working in our favor.
Mm-hmm. Mm-hmm. And if you can, actually, could you talk a little bit, because I know Dr. Sukal's background is quite extensive in terms of looking at sort of all aspects of veterinary vaccine development, from the vaccines to the adjuvants and other aspects of, as you were just talking about, the microenvironment. And I found it really interesting. And again, we can keep this sort of mid-range here. We don't have to go too far into the science, but one of the things that really intrigued me was that you were not just looking at isolating the sort of the tumor antigen profile, but you're really focusing on the way the microenvironment also of that tumor gets messed up. And I thought it was kind of a sort of a double whammy in the sense of what you're doing here, looking to obviously kill tumor cells, but at the same time modulate, immunomodulate the microenvironment. And there was this cool thing I was reading about small intestinal submucosa as an adjuvant. You don't have to go that far into that, but if you could sort of take us through exactly what's happening here with this vaccine, I think that's kind of cool.
Yeah, definitely. Well, I always like to talk about the small intestinal submucosa extracellular matrix component that we use as our microparticulate adjuvant. So if you say that 10 times fast, you'll be tired, and try writing that out a few times in a publication. You'd find ways to just make it an acronym. So what we're doing, and kind of what our technology was from Notre Dame, was, one, how we were preserving the antigens on the surface of that tumor cell. So what we wanted to do was really ensure that we're capturing the fingerprint of what went wrong. And when we think of cancer, cancer is a disease of mutations. It starts at the DNA level. It results in now these new mutational proteins that have been transcribed and now translated onto the surface of that cell. And if we take two patients with a glioblastoma, for example, about 70% of those proteins on the surface of those cells are unique to that individual and to that patient, because it's a direct result of the mutation that happened, and no two mutations can really be the same. So that means the fingerprints of both of those cells are inherently different. So what we want to do is capture that fingerprint at the time it went wrong, instead of trying to profile it out or even culturing out the tumor. When you culture out a tumor, you're selecting for the cells that are going to be best grown in culture. When you genomically profile the tumor and you're trying to get just certain peptides to be created, you might be missing how those peptides are actually folding, or maybe ones that could be better or worse T-cell antigens. So what we said was, let's keep it simple. Let's take the cell and let's take what went wrong, preserve those antigens, and now provide it back. Well, when you're providing something back, especially when you're providing back your own material to self, you need to make it look a little bit different. You need to rub the immune system in order to help stimulate the direction that you want to. So initially, we started looking at utilizing these cancer vaccines, of course, by themselves, but then looking at other adjuvants. So, like what, traditional adjuvants like alum,
Yep.
that
Yep.
again just provides this like, metal in order to just irritate at the microparticulate level, the response that you might be looking for. But the question that we had at the lab was, how can you truly drive the immune response into the direction? And what we ended with was this novel microparticulate called small intestinal submucosa extracellular matrix, or SIS-ECM. And why we landed there was that researchers at Purdue University had been working on this material, SIS. What they isolated was out of the small intestine, the submucosal layer or the middle layer is not only a highly immunogenic layer, has some nice tensile strength to it, but it's kind of where, the mucosal layer of all of our cilia and our intestines, it's going to bring in everything, all of the nutritions, all of the bacteria that we're eating in our body. And then it's going to everything's going to then get trapped inside of that submucosal layer. So there's a lot of immune cells that play a role there. And by being able to take that middle layer, decellularize it, lyophilize it, you now had a really great matrix to start promoting wound healing when it's applied as a sheet onto the body. So this has been used in millions of patients for hernia, for wound repair, for burn patients, diabetic foot ulcers as a medical device.
Mm-hmm.
And the question that they had was they had the ability of making this into like a microparticulate, like really grinding it up, getting it super, super small. And they were wondering, well, when we did that, what happens to the immune system? And they were able to showcase that they were able to drive a really nice and specific cytotoxic T cell response. So when you provided antigen alongside this microparticulate, you were able to drive a cytotoxic T cell response that was highly specific and can be replicated inside the body. So cancer vaccine, let's take the antigens, let's combine it with this novel material. Now we know we're driving it towards a cytotoxic T cell recognition. That's kind of like the 1 + 1 = 10 on the scenario. And we were able to really help multiply the response. So we think that it's a bigger and bulkier presentation back to the immune system. It's able to be brought back up into the antigen presenting cells really nicely, and it's able to nicely induce the T helper cell response to then downstream activate those cytotoxic T cells.
I love extracellular matrix. I just love the concept of it, and then it's one of those sort of frontiers. There's so much going on in it, and the fact that you can find these interesting bioactive components and ways to isolate them. I think it's totally fascinating and very elegant how you put the two together. Um, what we talk a little bit about you've been publishing you've published on sort of the safety evaluation of this process. You did the hemangiosarcoma study. You had some paper where you talked about I think so, like 5 years of retrospective analysis about cats. What is the what's the sort of the regulatory situation for products like that? I mean, I know obviously on the human side this has always been a major hurdle in terms of companies getting into or out of you mentioned some cases with regard to some cancer vaccines, but uh and ultimately how this all gets done, because clearly you got a lot of patients potentially out there. What's the FDA/EMA regulatory dynamic for personalized autologous
Cancer vaccines, yeah.
vet tissue vaccines?
Well, we'll say it's complicated, but it starts with the FDA Center of Veterinary Medicine.
Hmm.
They regulate your typical drugs or your typical pharmaceutical APIs. The USDA, the United States Department of Agriculture, they regulate all vaccines and all immunotherapies. So
Yeah.
anything that's driving a response on the immune system is going to be regulated by the USDA. So they do all of your rabies vaccines. They make sure that those are safe and effective effective. And
Mhm.
they also do PERS vaccines. They make sure that those are going to be safe and effective. Now, personalized and autologous cancer vaccines kind of goes against a lot of the structure that they previously had. So with companies like ours, and there's one other out of Kansas City, we've been working collaboratively with the agency in order to really understand the best way for these products to be regulated. Because at the same time, it's more so that your input of the patient's own tumor cell is inherently different. These are N of one vaccines, and they
Sure.
have to be for this scenario to work. We need to get those patient-specific antigens. But the manufacturing of the therapy is the same from start to finish, right? So there is no, or there should be extremely little deviation between the input and then what the final therapeutic actually is on the output. So, these are made in clean room environments. It's
Mhm.
made with, really strict, guidelines inside of hoods and how we're creating these therapies. We have strict quality control testing on the back end to ensure that we're providing not only a safe therapy, but also one that, has a potency behind it. So right now we sell our therapy as an experimental therapy
Sure.
for veterinarians that can be utilized. So safety and efficacy have not yet been established by the agency. However, we have publications and data, and we're running our last prospective study to help us get a special license. So it really is called a special license for autologous prescription products. So over the past, since 2013 until now, a lot has changed. It started with zero regulations in this environment, which is really scary for the end consumer. It then got very scary from the company side because they got like way overboard in terms of how they wanted to regulate these. And then, of course, with industry, we started working together in order to find a pathway to make it so that the consumer, the veterinarian, and the company can all have access to these therapies that are safe and are made in a very well controlled environment that have a potential for a reasonable expectation of efficacy.
Outstanding. Really outstanding. Um, I know we talked, obviously about, the sheer number of dogs and cats, but I also did notice that you recently published a retrospective study on 41 horses. Any interesting stories with regard? I mean, obviously, different type of patient, much bigger one.
Very
Than
big.
Yes, I actually, if it's funny enough, a couple careers ago, I was involved in a gorilla project, but I didn't have to get near the gorillas. Um, any interesting anecdotes with regards to creating cancer vaccines or extracting precursors for cancer vaccines from horses?
Yeah, I mean, when we're talking about some even in dogs, right? A lot of these tumors get a lot bigger than they ever would in humans because you may not notice it right away.
Right.
So you could go from like, a little skittle-sized tumor to a golf ball to a softball, pretty quickly in a dog with a soft tissue sarcoma, for example. Well, on the horse side, we're talking about football-sized tumors.
Mm,
And it is very hard to do a full resection of those cancers typically. I mean, it's not like getting a horse into a major tumor resection surgery is something that could be very easily done. So inside of our horse patients, very little tumor material is extracted in comparison to the mass that's there. But what has been really great has been looking at that objective response of being able to start seeing those tumors start shrinking or stabilizing in some cases, but being able to kind of turn that tumor back into the right direction and start seeing that regression has been really phenomenal. So we have a prospective study going on right now in horses that we're excited about.
hmm
And same with like different tumors in horses. We treat a lot of melanomas,
Mm,
squamous cell carcinomas, as well as sarcoid-based tumors. We've
okay.
been able to see some really cool responses that our team is excited about further exploring.
What you obviously, you as you mentioned, you treated about 4,000 patients. You've raised a nice amount of money, and I love hearing sort of these stories. Eventually, ultimately spinning out of the university, the entrepreneurial journey you've been on. I was looking at some numbers. It looks like we have about 127,000 veterinarians in the United States alone. Um, Clearly you have this, in terms of just getting to them all, scaling up. What's next on the agenda? I mean, are you looking to go it alone? I mean, clearly, from what I remember, sort of the vet pharma industry is more consolidated nowadays than the human pharma industry. But a little bit about what's happening next to take this from, 4,000 patients to 4 million patients, or whatever the business plan highlights.
Absolutely. And I think that's something that's still always going to be a work in progress, right? So there's a lot of consolidation within veterinary medicine that like your local everyday practice may have been bought by a corporate consolidator, which is good for small companies like me in the fact that I could potentially reach more hospitals with, fewer phone calls. But it still exists, from a fundamental business problem of, hey, like I have a really, really small footprint. I have a small sales team, and we need to be able to increase our, share of voice. So I think that in the future, being able to partner with some of the main animal health pharma companies would be the best way for us to really grow and expand the use of this therapy. And, I think it also just gets it into the hands of more veterinarians. So our goal is to, check off any last regulatory boxes, be able to continue growing our market share, and then evaluating really novel partnerships in order for us to help, grow it out even further.
Hmm.
But as a company, something that's really excited us has been, we took this notion of, hey, we have a technology that was originally positioned for humans, but we flipped the model and we were able to bring it into veterinary. So we
Mhm.
started evaluating other novel technologies that could have a similar pathway. And we've landed on a relationship with a really novel compound out of another university, and we're advancing that forward for clinical trials as well. It will expand the company outside of oncology, which
Mhm.
is not a bad thing.
Oh,
It has, better uh, kind of like market appeal for atopic dermatitis, which is a
nice.
$1 billion
Yep.
itchy dog market, for dry eye disease, for IBD. But there's also a potential to take the data that we've accumulated inside of animals and then flip that back up to
Mhm.
human. So for us, I think that there's some really exciting opportunities. And while my company, Torigen, will never be the company that could advance it to human, I could help lead at bringing together an expert team in order to do that, but
Yep.
leveraging all of our animal data. And instead of, the animal data being animal testing data, it's like, no, like that we are truly treating and helping client-owned animals in order to advance these therapies forward.
Mhm. Yeah, and then, that's obviously the dermatitis is huge. And then you get me thinking about all the other, because we don't think about it as much as probably should, but the things that cause us problems, diabetes, the autoimmune disorders. I mean, our pets get them all as well. So I think, yeah, I mean, your ability to based on your experience now in developing these novel interventions to branch out ultimately into these different therapeutic areas is just it is extremely exciting. I'd love to get your take on one other area, just because it's kind of nascent, but we see a few cases of clearly all these things that we're talking about, whether it's cancer, diabetes, heart disease, autoimmune. A lot of it's connected to aging. And we've seen a little bit of activity in the vet space in terms of developing specific drugs for pets that potentially interfere with biologic aging processes. Anything interesting on that front, or do you look at that as a potential target for your company one day, or is it kind of a niche right now, in your opinion?
Yeah, I mean, it's definitely something that I'm learning a lot about, especially as of late. So a dog is going to age 7 years faster than 1 year of a human life, right?
Yeah.
So we automatically know that. And bigger dogs have a even shorter lifespan than smaller dogs. So I think being able to have these compounds that could potentially extend lifespan. I mean, some of the chatter on, Facebook has been like, I would do anything to have my dog live longer.
Mhm.
So, I said I started this by saying I would walk across the earth, if it meant helping and saving my dog. And I truly do mean that. But I think when we look at, well, what does it mean to have a commercializable and ready product in the veterinary space? I can say that I'm going to walk across the earth, but when it comes down to the brass tacks of, okay, what does this drug do in order to help my dog live longer? I think that's going to be the question that we're going to look out for.
Yeah.
So we also know that 50% of all dogs are overweight.
Yeah.
So if you're able to reduce weight or create an Ozempic for dogs, those dogs may live longer, right?
Right.
But is that a age related biological target, or is it a factor of metabolism that we're able
Right.
to
Right.
start
Right.
reversing? So I think that's going to be the question that will definitely come out in regards to the commercialization piece. Because if a veterinarian is like, well, this is just a weight loss drug for dogs, I have, generics that are able to do that just as well. Or is this truly a novel factor that is going to allow these dogs to truly, extend their aging profile and increase their telomeres? Like, I think
Hmm.
that that's
Hmm.
going to be something that's going to come out over the next couple of years. And then will I, as the consumer, put my money where my mouth is in order to say, yes, I want this drug to help my dog live longer?
No, I appreciate your insight in that. I really do.
But it's super
Um,
cool. And I mean,
yeah.
they've been able, the one company that's out there, San Francisco based company called Loyal
Yep.
for dogs, they've just closed another 50 million. I mean, as a woman entrepreneur, I know she is another woman CEO. I give nothing but extreme respect because this is a hard world to live in, regardless of your industry sector.
Yep.
And, I wish nothing but the best.
Yep. Yep. Ashley, what else is coming up for you and the company in 2024? I see there's a few sort of veterinary medical association meetings that you're getting to. Anything else on the calendar that we should know about in terms of conferences, different talks you may be giving, places that we could run into, possibly meet you? Anything that I missed, please.
Yeah, totally. I am leaving tonight for West Virginia.
Nice.
I'll be down at the West Virginia Veterinary Medical Association this weekend. I think a few members of our team are going to a San Diego Veterinary Medical Association. So we do a lot of the local VMAs, especially where we have speaking opportunities, because for us, we aim to educate general practitioner veterinarians on our product and the ability to potentially treat cancer in their practice, because there are only 400 veterinary oncologists nationwide who do chemotherapy on a daily basis. So going out to somewhere like West Virginia, there's really very few opportunities outside of the University of, Tennessee and North Carolina to get your pet treated after a cancer diagnosis. So
Hmm.
being able to educate general practitioners that options out there do exist really help spread our share of voice too. And we have two oncologists full time on my staff. Our chief medical officer is a veterinary oncologist. Our vice president of veterinary affairs, veterinary oncologist. So it becomes like we have resources available in order to help these veterinarians when considering, is immunotherapy the right option for me at this time. Outside of that, I have been traveling for the past couple of months, raising capital, setting up some really great strategic partnerships, both domestically as well as internationally. I'm excited to be home for a little bit, but I'm always available in order to talk to anyone, especially if we want to just talk science over a cup of coffee. And anyone could reach out to me and the company, info@torigen.com, and it'll get to me.
Awesome. Awesome. And we will obviously put the link in the bio of the show. But really exciting stuff, Ashley. I really enjoy hearing stories, success stories like this of entrepreneurs like yourself that have have made it. I mean, I clearly look at this as a success story in biotech, and obviously in an industry that has such a high failure rate. I'm really rooting for you, and it's gonna be fun to continue to follow you and the company, and hopefully do a follow up maybe next year, see where everything's headed. Again, for everybody that is going to be listening to this particular episode of our show across the various podcast networks or watching on our YouTube channel. Again, you've been spending time with Ashley Kalinauskas, founder and chief executive officer, Torigen Pharmaceuticals. Ashley, I really want to thank you for taking the time out of your schedule to talk to us for a little while, tell us what you're up to. Obviously, thank you for what you do. And as we like to say here on our show, thanks for helping to create a better tomorrow for us and our pets via what you're doing. Really great story.